Okay, welcome to Laboratory Medicine Grand Rounds. I'm Brian Schertz, the course director for this. So we have a little bit different Grand Rounds this time. Instead of one speaker, we have 6. And so this is an interesting one because we wanted to introduce the Department of Laboratory Medicine to the Brotman Baty Institute, which is a large, complex organization. I'm not going to introduce all the speakers because I went through all of their training and resumes, then it would last the entire hour. So we're going to start off with Heather Mefford, who's the associate director, is that, of the Brotman Baty Institute, and she'll explain what the Brotman Baty Institute is for us, and then we'll gradually hand the microphone off from one person to the other person, and you'll hear many different parts of the Brotman Baty Institute, and perhaps not all of them. So I'm just going to hand it off to Heather, and we'll get going.
Great. Well, thanks, Brian. It's great to be here, and I think we have a nice lineup to give. Our goal today is really to just give you a broad overview of some of the activities of the Institute, which some of us will refer to as the BBI, and to give you an idea of perhaps at least some inspiration for thinking about how you can get involved and what sorts of projects we're excited about supporting. So the BBI was launched about 1.5 years ago, and it was with about 1 year or 2 of planning prior to that, and was made possible by a generous gift from 2 families, the Brotmans and the Batys. It was a $50 million gift that was intended to be used over about 5 years for the development of a precision medicine institute. The donors and the leadership are all committed to the fact that this institute is designed to be not a University of Washington institute, but to involve faculty from the University of Washington, from Seattle Children's, and from the Fred Hutch. So kind of a cross-institution institute. The overall goals are really to accelerate development and application of various approaches, some of which you'll hear about today, in a clinical setting. There's a strong emphasis on fostering basic science, developments and discoveries that will lend themselves to translation into precision medicine or to studies that will help us better understand specific disorders and pathology. And as I already said, we really want to leverage the strengths across Seattle, so involving individuals from all 3 institutions. As I said, we launched we actually opened for membership a little over 1 year ago with the goal of having 150 people interested in signing up, and we've met and surpassed that goal and now have over 230 official members of the BBI. And I'll come back to this at the end, but this is meant to be an inclusive, not an exclusive institute, so you can talk to any of us afterwards about how to get involved if you're not already. The members come from all 3 institutions, as you can see by this pie chart here, and the proportions roughly represent the size of the various institutions. So the structure of the BBI is such that we are governed by the CEOs of the 3 institutions. So Paul Ramsey from the University of Washington School of Medicine, Jeff Sparing from Seattle Children's, and I just blanked. Gary, I was going to say Paul, Gary Gilliland from Fred Hutch. So they are really our governance committee. The scientific director is Jay Shendure from Genome Sciences, and then I am the deputy science director. We also have a lot of input from the various communities. We have a scientific advisory board from outside scientists who help kind of make sure that we're on the right track with directions we're thinking about going. We have a management committee that's made up of faculty from all 3 institutions here. We meet on a regular basis. And then we have a community advisory board that we started meeting with to think about things like long-term sustainability, branding, and those sorts of things. The way that we've set up kind of the activities of the BBI are divided into what we've called platforms, programs, and flagship projects. And then in the past year, we've added a new project that's really a beast of its own called the Seattle Flu Study. The platforms are intended to be resources for BBI members to A lot of it is data generation, and they're supposed to be accessible across all 3 institutions at a uniform rate. So this is something you don't experience for all platforms like that. But these include some you're going to hear about today: the BAT Lab, Precision Diagnostics, Single Cell, Bioinformatics, and then we have several programs that I'll just outline here in a minute quickly that are made up of individuals who've helped us kind of shape what direction we want to go with these specific activities. Some flagship projects. We had our first round of funding last summer, including funding in the funding with data for generation of single-cell sequencing data, as well as funding for some small pilot projects that we called catalytic collaborations for individuals who were working collaboratively across 2 or 3 institutions on a new precision medicine project. And Colin will talk more about that third one there. And the flu study I'm not going to touch on today, but we're happy to answer questions about that afterwards if you'd like. These are some of the things that you're going to hear about today and the people that are involved, some faces that you might recognize. We're going to hear more about the BAT Lab and their single-cell sequencing projects. Tina will talk about CellFree DNA applications. You'll hear from Kelly about an initiative to create a living biobank. And then some things we won't touch specifically on are that we have a large-scale sequencing center and are working on setting up a proteomics platform. So just to let you know, the working groups are really groups of faculty that we got together and said, where would you like to see precision medicine go in Seattle and what should we focus on? We had several meetings over the course of the first few months and then are following in the lead of the investigators. And this is just to give you a flavor of what the approaches are, what the topics are, who the leadership of those groups are, which you'll see come from, again, all 3 institutions, and then what the ongoing work is for each of those. Most of these you'll hear about in the following talks. The Pediatric Genomic Sequencing Working Group is one that we're working with Lab Medicine and the Northwest Genomics Clinical Lab to advance clinical exome sequencing here in Seattle. And I think mutational scanning may be touched on briefly as well. So I'm going to hand it over to Leah Starita, who is leading the BAT Lab, which she will tell you all about.
Hi. So the BBI Advanced Technology Lab, which is we lovingly call the BAT Lab, is going to be a fee-for-service kind of core facility. We're kind of calling ourselves like a 2.0, Genome Center 2.0. So we plan to provide support for multiplex functional assays, and I'll tell you a little bit more about that. Single-cell genomics, so single-cell RNA-seq and possibly ATAC-seq in the near future. And on top of that, importantly, a platform for analyzing and visualizing that data. And we also, through this flu project, became a high-throughput extraction facility. And we also have a high-content imager available for use as well. Okay, so these multiplex functional assays have been developed in the genome science, mostly developed in the genome sciences department over the last couple of years. And what's really great about them is that recently, with all of the sequencing that everybody else does, they seem to find variants of uncertain significance very often in a lot of the sequencing data. And it would be great to be able to do a functional assay to understand the effect of that variant of uncertain significance. However, doing them one at a time for things like BRCA 1 and 2, where there's thousands and thousands of these, is kind of time-consuming, right? So by developing these multiplexed assays, we can look at hundreds to thousands of these variants in a single experiment. And although they're all different for different functional elements like promoters, enhancers, and proteins, they all consist of a few key steps, and we are hoping to be able to provide support for these. So for example, we have to make all the variants we want to test, and we usually test every possible variant in a sequence. So we make these types of libraries, and we can we can make these types of things for you. You have to get those assays or get those variants into cells, and we have worked with many labs, including the Fowler Lab in genome sciences, to develop, or they've developed landing pads to get the proteins in, and also we do saturation genome editing, so we edit those variants right into the genome. And then functional assays, so we've developed many ourselves, and we plan on supporting like a kind of a consult for developing functional assays for everybody's favorite gene of interest. And then, the readout for these types of assays is next generation sequencing, which is what allows our throughput. And we can also support amplicon sequencing there as well. And from these assays, you wind up with sequence function maps to tell you exactly what the functional consequence of your mutation was. Okay. The next thing we're going to be doing is single-cell RNA sequencing. So in this case, people with interesting samples, whether they be from a diseased tissue or a certain time in development or even model organism, can give us pieces of tissue or cells, and we will be giving them back single-cell RNA-seq and possibly ATAC-seq data. So the way we do this is by combinatorial indexing, and we're now able to do, this was developed in Jay Shanduri's lab by an extraordinarily talented grad student named Jun Kao. And we're now able to look at about 2 million cells. We're able to profile the transcriptome of 2 million cells in a single experiment. Okay, and so what I'm showing you here on this slide is just a t-SNE of all 2 million of those cells, and each one of these little clusters is a different cell type. And importantly, this platform is more useful because we actually have a bioinformatics platform associated with it, right? We've been really lucky to recruit expert computational biologists and data visualization and web developers to this platform. And and what I'm showing you here is just a screenshot of our web tool, our visualization tool, and it can actually be found on the brotmanbaty.org website, where you can actually kind of play with this data. So this is, what are they? These are connective tissue progenitors, but if I wanted to go look at the intermediate mesoderm, I could go through a drop-down menu and find those, and that's where these are. These are progenitors of like the kidney, and you can see here all of the differentially expressed genes in that cell cluster. And this platform is actually like kind of the most advanced right now in the entire field, which is really cool. And we're hoping to be able to populate a bigger data set using the RFAs, or the data generated through these RFAs that Heather told you about. We have about 30 projects, anywhere from trying to figure out what the cell of origin is for things like Joubert's disease, down to human fetal tooth development and regeneration in zebrafish fins. So we've got a whole bunch of different projects going, and right now we're trying to scale up or move the single-cell RNA-seq from a grad student's project up to a production level experiment, and we're also still learning how to deal with a lot of adult tissues, which apparently have a lot of RNase in them. But we're getting there, and we're about to do our first production run, so we're pretty excited about that. Because of the single cell experiments, we had to buy a high throughput solution to actually counting cells, right? And so we wound up kind of buying a high content imager, which seems like, killing a mosquito with a cannon. But what's really nice about this is that we can actually we can offer time on this instrument as well. It's a pretty cool little machine. We can count 96. We can really accurately count our nuclei that go into the single-cell experiments, but we can also offer this to other people, and it does fluorescence in 4 different colors. It does bright field, and so it's a Molecular Devices Pico, so if anybody wants to use this thing, you can contact us. Okay, and the large-scale biological accessioning and nucleic acids extraction. So through the Flu project, we have purchased a Roche MagnaPure 96, so we can do 96 RNA or DNA extractions per hour. And this is research use only, but we can definitely do that on a fee-for-service basis as well.
All right, so I'm Colin Pritchard. I think most of you know me here. So my role in the BBI is as the head of one of the platforms, which is the Precision Diagnostics platform. So this is the kind of the clinical lab testing arm of the BBI, and I think you'll get probably hopefully everyone's already got a flavor that this is just one piece. I mean, the BBI is mostly a research initiative, but this is the sort of the clinical lab testing arm of that. So what is the Precision Diagnostics platform? So it's administratively housed here. There are member labs. We're trying to recruit more member labs to be interested in being part of the network. There are small pilot grants. So the Heather Mefford already referred to the Precision Medicine Lab Pilot Awards, and we've already given out one round of those. So it's a nice opportunity to know about. There are both faculty awards and trainee awards. These require that at least one person involved is a CLIA lab director and system. So these are really designed for true clinical validation and development projects. There are multidisciplinary variant review teams. So this is sort of building on the model that we've had going back now 8 or 9 years, where we look to departments across our 3 institutions for content expertise in both somatic and Mendelian genetics and build review teams around that. Molecular tumor boards, we have at least 4 now, going on 5. There's assay support for BBI working groups. So Dr. Lockwood will talk about the CellFree DNA working group, which is basically housed within the platform and basically taking advantage of the work of the Genetics and Solid Tumors Lab in building CellFree DNA Diagnostics and then harnessing that for research. There's the clinical applications of research and emerging technologies. That's a mouthful, the CARET unit, and that's what Dr. Schertz will talk about. That's his role. That's also housed in the platform. So what are these pilot awards? Some of you, I'm looking, are actually awardees, so you know what these are, but not everyone does. So they're awards for primarily validation of assays and implementation. We recognize it's really a gap here, so there are a number of pilot award mechanisms in our system, but they tend to favor researchers even when the stated goal is for clinical assay development. And so we really want to sort of get that money seeded to actual clinical lab directors for these projects. So that's the goal. The other gap that we noted was in trainee funding. So we hear this from our trainees in pathology and lab medicine, but also trainees in other disciplines and other areas that are interested in molecular diagnostics, that there just aren't enough awards aimed at them. So what we have is 3 to 4 faculty pilot awards, $50,000 each, and 5 trainee pilot awards, $10,000 each. So that's a lot of trainee awards for a relatively small pool. So for the trainees in the audience, I really want to emphasize this is a nice opportunity. There'll be there presumably will be another round coming up. Probably won't go out for another 6 months or so, but just to be aware of, we did get a lot of nice applications this round that we were able to fund. And then support for implementation of new technologies in BBI-affiliated clinical diagnostics lab is what the goal is here. So here's just an example of a few of the variant review teams. So you can see these are large teams. We have teams around cancer predisposition, somatic cancer therapy. We have teams around pediatric syndromes, which I'm including exome sequencing in there, and then also inherited marrow failure syndromes. And we're building new teams all the time. So the goal is really, again, who has content expertise in our system? Doesn't have to be someone in pathology or lab medicine, just anywhere, in some field, and then let's build an assay around that. That's where we're going to build our expertise, and let's bring that person or persons into the team. And we on the clinical lab side can help with how we do the actual clinical lab process and sign out and billing and all the rest of it. And take advantage of that content expertise in that variant review team, and that really distinguishes us in terms of expertise. So we have a test catalog for the BBI now too. So this is built upon the online test guide. So those of us in lab medicine know what that is. Those of us not might maybe not have heard of that. So we were thinking about building out a brand new test catalog, and then I was talking with Noah Hoffman, and he said, who's our informatics division head, and he said, well, this is silly. We already have an online test guide, and we could just build an API to it. So that's what we've done, and so that's about to launch. And so the goal is then to sort of take tests that are already in the standard format in online test guide, and then again reach out to member labs and facilitate, if they're interested, getting their test menu into that same format, so then it can be linked out to the website. So here's just an example of what that looks like. That'll go live on the site, I think, fairly soon. I was just talking to Nola Klemfus about that. So for those of you who are in clinical labs or clinical lab director where you think, hey, I have my lab, my test menu, I want that to be on the BBI website so that more people can have visibility to order it, talk to me at some point. We're happy, or to Brian Schertz, who's leading that. We're happy to link those in or go through the process of how you do that. The molecular tumor boards, currently we have a genitourinary precision tumor board. This is primarily focused on prostate cancer, now extended to bladder cancer. We've been doing that since 2013, but we've really leveraged the BBI infrastructure to expand this out more. A pediatric precision tumor board led by Tina Lockwood, Bonnie Cole, Sarah Leary. It's been going on for a couple years, and most recently a GI precision tumor board led by Eric Connick, Stacy Cohen, focused on mostly colorectal cancer, but other GI malignancies. So we want to partner with the Seattle Cancer Care Alliance to really amplify these molecular tumor boards and think creatively about how we can harness this collaboration of BBI to go even bigger with these. So we have a really nice infrastructure in place. Can we do community outreach? Can we leverage all this expertise within the BBI and our institutions to then drive referrals to our centers? You know, there's a lot of nice synergies we can think of there, while also doing some nice education for the community. We're also thinking about a Mendelian molecular board. So this, of course, wouldn't be a tumor board, but you know, how do we do that, and can we do community outreach around Mendelian boards as well? So interfaces with the precision diagnostics platform and other aspects. So this is a really busy slide, but the point of this is that these platforms and working groups that Dr. Mefford described at the beginning, they really are all interlinked and synergizing. And so for the precision diagnostics, clinical diagnostics platform, there's already very meaningful collaboration with large-scale genome sequencing through, for example, the All of Us grant, which we've partnered with the Genome Center to sort of land. We're one of 3 genome centers in the country that have landed this large grant, and that's, you know, a direct, I think, success of the BBI and that collaboration. We just heard Leah talk about the BAT lab. We're excited about the collaborations there and how these sort of research platforms how can we sort of get a pipeline from clinical testing to functional tests that maybe in the research setting. How do we do that, you know, for patient care in a way that works? And I think we can pull it off. Anyway, and we'll hear a lot about the CellFree DNA working group and the assays around that Dr. Lockwood's going to talk about next. The CARET unit, Brian Schertz is going to talk about now, so I'll let him do that. Okay, so.
Thank you, Colin. So this is a very small part of the BBI, as you can tell, but we wanted to talk about it specifically with this audience because it's maybe one of the pieces of the Brotman Baty Institute that's most relevant to laboratory medicine. So, so what is this? This is something actually that I think John Tate was the first person who thought of this, and we've proposed the BBI, and they said, yeah, this is a good idea, we should do this. So the concept behind this was that many people in laboratory medicine have researchers approaching them saying, hey, you run a clinical diagnostic laboratory. I have this great idea for a clinical diagnostic test. Can I pitch it to you? And how do I get this to patients? And sometimes they're phenomenal ideas. You think, yeah, this is wonderful. We need to be doing this now. Other times, they're ideas that need a lot of work before they're ready for patient care. And that type of clinical consultation, or that type of diagnostic service consultation, is something that the people who know you don't realize how lucky they are to know you. And there's many people in the university who aren't lucky enough to know someone in laboratory medicine who has skills to develop a clinical diagnostic test. And so the thought is that we should be providing these services. This is what we do. This is what we love to do. We should be providing these services in a more structured way so that more people can access the knowledge and skills we have here in laboratory medicine. And this would be something that would be really unique. We see so there's this need. We have the resources to be able to do this. And this is something that other components like ITHS or Comotion or technology transfer offices really don't have the skills to do, because oftentimes diagnostic tests are not patentable and don't lead to immediate investment from large companies, but that could meaningfully improve clinical care. So the purpose is to help translate new technology into clinical tests. So what the CARET directors, the CARET consultants would do would be to evaluate consultative services, decide whether they're ready for prime time or not ready for prime time, and then provide feedback to the individuals who are interested in developing technology and moving it to clinical diagnostics about whether what the next steps are, and then potentially shepherding those clinical tests or the ideas that are ready to move on into our laboratories, into the laboratories at the University of Washington Laboratory of Medicine or at Seattle Children's Hospital or at Fred Hutch, which has CLIA-approved laboratories that are really for research testing for pilots. So we would support that. The resources that we have or that we're putting together right now that we're developing are a guidebook and a framework for consultants, assistance with internal memorandums of understanding. If somebody from University of Washington comes, you can start working with him with just a simple memorandum of understanding. Potentially assistance with external consulting agreements. Oftentimes, someone will come to you and they say, I'd like some help with this diagnostic test, and they may or may not tell you that they're already starting a company. So hopefully we'll weed those people out and we'll be able to help you get consulting agreements with them that make sense. And then also connections with the Institute for Translational Health Sciences, for the tech transfer offices at the different organizations, and so that people who come to them will know that there are resources available even if they don't have an idea that is patentable or that leads to the technology that they're thinking about but may improve patient care. They can send those people to us, and we can help them make the most of their diagnostic technology. Hopefully those connections will also allow more people to be able to get better ideas to patients quicker. So these are the people that this is a short list of people that I've already spoken to about this and who have agreed to be CARET consultants. The only reason why I put this list up here is that if you are a director in a CLIA lab in laboratory medicine and want to be a consultant, or perhaps in pathology want to bring clinical diagnostic tests as improvements to patient care, and you want to be on this list, then please come talk to me, because we can use the more people that are available as consultants, then the easier it will be to pair the diagnostic testing methodology with the expertise that we already have in laboratory medicine. So please come find me afterwards if you have questions, and now I'm going to hand the mic off to Kelly Smith.
Thanks. I'm Kelly Smith. I think most people here know me. So I am one of the leaders of one of the youngest working groups in the BBI, and that's the Living Biobank Working Group. And just as a little background, the name Living Biobank is essentially stolen from the person who popularized it, who's Hans Clever, who's the person who put organoids on the map and is the individual that founded the Hubrecht Institute in Utrecht, Netherlands. And so the organoid technology is just exploding, and this has been used to develop models of normal human tissues, and also more recently been used to develop models of tumors as well. And this is just one of many papers showing the development of multiple organoids from breast cancers, and they show that they have over 100 breast cancers, which they developed nearly 100 organoids, and all of those organoids essentially have very similar characteristics to the actual tumors that they're derived from. and they were to be able to make these from all sorts of different tumors. The goal then, in their large enterprise, is to actually create this organoid resource, characterize those, and then be able to distribute those to researchers. And I thought, and that was sort of the overlying goal of what we're trying to get started here, is to create then a resource where we can preserve fresh, viable human tissue and tumors so that they can be used then for researchers to do basic translational preclinical studies, as well as develop potential functional diagnostic tests and test hypotheses. Some of the utility of these organoids is shown here, where they actually, in this paper, show how they looked at the ability of different inhibitors of the HER2 pathway and the EGFR pathway to inhibit the growth of different tumors. And notably, there's a couple of, I guess my arrow's disappearing. Oh, there it is. There's a couple of tumors here that have behaviors that are not predicted based on the normal HER2 staining. So you predict that something that's HER2 positive would be inhibited by low levels of drugs that inhibit this pathway, but there is one over there, 68T, that doesn't, and there's another one there on the right-hand side that is at least partially strongly inhibited. In addition, in looking at this data, which you might be able to see, is that there's actually a bunch of heterogeneity within these actual tumors, that instead of being a single solid rectangle, that those rectangles are divided up into smaller boxes. And so there's a lot of heterogeneity in tumors that can be essentially revealed by studying the actual organoids themselves versus just studying the tissue in paraffin sections. And similarly, looking at diseases from patients, they can actually see that the organoids that are developed from the patients actually respond similarly to the tumor within the patient. So you can predict biology that might be happening in patients. And so these are then some of the examples that have been brought forward to show that this is really an essential tool, I think, for us moving forward, to be able to have a resource where we can use viable human cells that model human disease to do these types of basic translational, preclinical, and actual clinical studies. So currently, we see specimens flow through our labs in many different ways, but the vast majority of them go in through the clinical lab, and then there's the residuals here that are then, some of those are repurposed and used then, stuff that's not used for clinical care, for research studies. And then most of the fresh tissue that's used for research actually goes then out and dispersed into individual labs. The goal then for setting up this living biobank was actually to try to create a mechanism where we can actually help preserve some of this fresh tissue in a manner that's accessible for researchers to be able to take advantage of and use viable human tissue to develop models of human disease. So what sort of components do we envision as part of this living biobank? Is essentially it'll help receive and process these fresh samples. There'll be some quality assessment and control of the samples, as well as annotation to include experimental and in clinical data, and then a local mechanism to store and distribute these types of samples. So what have we done so far as part of the working group? So the working group has had 2 meetings to discuss this project. The leaders of the group include individuals from Seattle Children's, the UW and Fred Hutch, so Bonnie Cole, who's in pathology at Seattle Children's, David Bird in surgery from the University of Washington, Eric Holland from the Fred Hutch and STTR, and myself. Recently, we had an RFA for some pilot grants to support the collection of viable tissues, to develop these model systems, and to generate best practices and standard operating procedures for putting together this living biobank. This RFA has ended up funding 4 projects, and we've also had then our first startup meeting for the group where we began discussing these. And so the first outcome is really to set up the charter of what do we hope to accomplish as this group. And the charter for this group is to develop best practices for biobanking and propagating viable tissues from cells in patients, to develop a working model and plan for this resource, to develop a critical mass of local expertise so that we can help the greater community enter into this field and engage the community as well to help contribute to and be part of this project and effort. And finally, also, one of the more important areas is really to provide strategies to help ensure the growth and sustainability of this resource. So that's where I'm at, and I think I'm going to move on to Tina.
All right. So my uncle actually, he was a musher in the Iditarod. And so there's an award in the Iditarod if you were the last finisher, which is the Red Lantern Award. So I officially am in pink, but I received the Red Lantern Award today for talking about CellFree DNA Diagnostics. So this is similar to what we've already talked about, an initiative that involves leaders from UW, Steve Henikoff at the Hutch, and then Sarah Leary at Seattle Children's Hospital, Seattle Children's Research Institute. We have a bioinformaticist for the working group, Jonathan Reichel. If you have been in NW 120, he sits over near where the genetic counselor sits. So if you're wondering who that person is, I'll point him out on another slide. And I particularly want to thank the program management and support that we have provided by Nola Klemfus, Hart Edmondson, and Moon Chung has really made sure that we have all these projects going off without a hitch, which is sounds like it's not that hard to do, but I'll show you that we actually have over 600 samples that are coming through as part of this working group, and that's hard to logistically coordinate. So huge thanks to Moon. And if you are not familiar with CellFree DNA, I feel like that analyte has entered our vernacular, but here's a representative image, which is to say that what we're taking in the sense of cell-free DNA, it's just a DNA that's freely floating and in a fluid type. So that could be, we're looking at specifically in plasma, so from whole blood. We could be looking at cerebral spinal fluid. We could be looking at urine. Take a fluid sample, and so far we've looked and we've been able to detect cell-free DNA in it. In this specific image, we'll come back to it later on, this is actually from a paper out of Jay Shanduri's group. Matthew Snyder was the first author, and this was looking at tissue of origin, which again I'll come back to in a moment. But really one of the reasons to have this working group is because we have a lot of expertise locally within cell-free DNA. So not just in lab medicine, I'll show you a little bit of what we've done in lab medicine, but in genome sciences, in pathology, at the Hutch, at Seattle Children's Hospital, really across the institutions. And we really want to leverage that because we see this being a very promising application as we go forward in diagnostics. So I'm going to show you what our assay is for cell-free DNA. And while I'm very proud of our assay, while I'm showing you this is not just because I'm very proud of it, but that's actually, this is the assay that we're using to fund our pilot studies for the cell-free DNA working group. So this is work that particularly Bryce Colbert, a lead in our NGS analytics group, has developed with me and Steve Salaponte and the rest of the genetics and solid tumors lab. I'll show you a picture of us in a moment. But why we developed this assay was actually for prenatal, initially aneuploidy screening. And then we've extended beyond aneuploidy to just subchromosomal events, so either gains or losses. And what we're looking at here is a plot. This is from a pregnant woman with an XY fetus. The fetal fraction was 5%. And what we're looking at up here, this is a chromosome 21. We're looking at just it's basically a copy number plot. So this black line is showing us a copy neutral state. So if you have 2 copies of, for example, chromosome 21, that's what it would look like. If you look across the whole genome, down here we're looking at all of the autosomes. So chromosome 1 over here, and again, here's a copy neutral state. We have chromosome-specific cutoffs that we use. Chromosome 19 is naughty. It has very wide bars because it's often, we see a lot of copy number variation in chromosome 19. So that's what we see, again, in a normal pregnancy. Here's an example of a trisomy 21 XY fetus with a fetal fraction, again, pretty low. So 95% of the cell-free DNA that we obtained in the sample was from mom. 5% of it was from the fetus. But because we have put in some very specific limits, you can see here, it's not, your eye isn't really drawn to that as being a gain, and that's the power of course, statistical tests. here we can see that we have, even with only 5% coming from the fetus having a trisomy 21, we can very clearly see that that's abnormal. While cell-free DNA is actually revolutionary in what we are doing for prenatal aneuploidy screening and sub-aneuploidy screening as well, what we wanted to do in the context of the working group was actually to leverage this assay because it's a very nice standardized assay. We get samples in a standardized way, we extract them in a standardized way, and then we put them through a common workflow. So I'll show you an example of when we had something that we didn't expect to see. So this was a pregnant woman. And so during her pregnancy, what we saw was that we had a lot of copy number variation that was present in this patient that we did not expect to see. So here on chromosome 17, there's a significant gain. So this is a statistical test, so how close you are to the mean. So we had a z-score up to 60. So I think that's, 60. And so what that actually corresponds to is an amplification of ERB2 and HER2. So what we were thinking is one possibility is that this could have been a breast cancer, but you can see HER2 amplification in other types of cancers as well. What it very nicely showed, though, so even though you have this significant elevation here on chromosome 17, if you look at the whole of chromosome 17, it's actually not increased, right? Because 17 is fairly large, and you only have this very small region that's amplified. So what the advantage is of this assay is that we can see both these focal gains and losses, as well as whole chromosome gains and losses. So here we have an amplification, for example, of chromosome 1. I think that's chromosome 8. We have a deletion of chromosome 9, and so on and so forth. And this is really the reason that we wanted to use this assay, because it's fairly agnostic to the sample type that we're using. So that was what we put out our pilot awards for. And I hope for forever this is the situation in my life, which is to say that we funded everything that we could, so everything that was feasible within the confines of the assay that we were using, so our copy number variant assay. 15 pilot awards were in fact funded. So that represents over 600 samples, 630 samples. And the projects that we're funding really range across the entire spectrum. So some we're only doing the handling and the extraction for, but then we're also looking at placental biology, allograft rejection, oncology, organ damage, mutational signatures. Put up your favorite project here, so that could easily fit into the mix. So we funded these awards, I believe, in January of this year, and so the expectation is that by March of 2020, we'll actually have we'll have completed all the testing for these 630 samples. Nobody in genetics and solid tumors, have a heart attack, but we do have a lot of samples that are coming through the lab. We had our first symposium, April 17th, and brought together all of the members of the working group who were able to attend, and that included some of our keynote speakers, as shown here, some of whom you'll recognize from lab medicine and beyond. And then told you I would tell you about Jonathan. So here's a picture of Jonathan Reichel, our bioinformaticist. Obviously, the computational analysis is fairly challenging in this space, and Leah mentioned as well within the BAT lab. So that's our current state, is really to finish all of these pilot awards that have been awarded. And then moving forward into the second year of the working group, what we really want to be doing is to be taking this a step further. So one reason that we've used this generic type of a platform where we're doing low-pass whole genome sequencing to determine copy number is that we can apply that later on. What we really want to be able to do is to tell you, in this gamish of cell-free DNA that we get out in this context from plasma, where is it coming from? So what is, in fact, the tissue of origin? You can imagine how powerful that would be if you think of a cancer of unknown primary, if you think about sometimes in the context of placental biology as well, is there an abnormality that's actually going on within that placenta? So that's what we're looking to do now in going forward into the second year. And then we would actually be able to leverage all the data that we've already generated. So if I want to have a normal control, for example, for my oncology study, I would be able to use for maybe some women that were pregnant and there was not an abnormality that was detected in that pregnancy. And so I want to thank, in particular, the Genetics and Solid Tumors Lab, who are actually doing all of these studies. And I'm going to point out Bryce, who's a floating head in this picture because his family had recently had a baby. So thank you to lots of different people, and I'm going to hand it back over to Heather to wrap up.
All right, well, I am amazed that we are on time. And we're going to have time for questions. So I'm just going to wrap up. This is the front page of our website, so it is brotmanbaty.org if you want to go visit the website. There's information about all of the working groups and platforms there, as well as contact information for many of us. And if you're interested in becoming a member and you're not already a member, then you can go to the website or you can come talk to us when we're finished here. Tina mentioned that they had their first symposium for the CellFree DNA working group. One of the things that we're trying to do is highlight the work of each major effort through these symposia. And there are 2 that are coming up. This is also available on the website, but I wanted you to know about them now as well. The next one is coming up quickly. It's the single cell symposium on June 3rd. And then we plan to have one in the fall for the pediatric sequencing working group. And I'm going to stop there. And if you have questions for any of us, everybody's available to answer questions. And please use the microphone if you have a question. Thank you.
What, in terms of the stuff you were describing, taking these tissue samples and so on, how does this relate to, if any way, to Northwest BioTrust, which many of us know and don't particularly love?
So the only relationship to Northwest BioTrust is through tissue procurement. So tissue procurement is handled through Northwest BioTrust in the UW facility, and it's handled through the Seattle Children's. They have their own acronym for their own lab, and so that's the thing. The procurement will go through the same routes, but the idea is to have the community define really what this entity will be and how to run it and how to govern it.
I think many of us are overwhelmed actually by the potential here, so we're stunned. But I'm wondering what you, of the group who've been thinking about this, what do you think is the low-hanging fruit for CARET, for translation into diagnostics across the campus? What do you, who would you seek out, and what have you been thinking about so far?
Thanks, Mark. I think that question is for me. So I think that's one of the reasons why we need this carrot, is that, you know, in order to, I guess you can use the carrot and stick analogy, we want to attract people who have good ideas to us, and we really don't know what the low-hanging fruit is for the next big thing for diagnostics, or even actually what this is probably best for is not the next really big thing, because the next really big thing is probably going to and get, you know, massive amounts of investment from Silicon Valley or from wherever. But the next thing that's really good that is hiding somewhere. And so I think that this is we don't really know where that's coming from. This could be coming from an undergraduate up on Upper Campus who has a great idea for a new chemistry, or it could be coming from a clinician in the OB-GYN department who doesn't know anyone in lab medicine and thinks, you know what, if we could just test the tissue a little bit differently, or if we could just, you know, I read a paper about an analyte, and if we could just test that analyte, or if we could figure out a way to combine these two tests together, then maybe we could improve care for a specific rare type of disease that people are ignoring. So those are the type of people that I hope that will hear about the CARET service and come to us and say, you know, I have this idea. It shouldn't be too hard to do. How do we get this to patients? Right now, when that happens, sometimes researchers start their own, you know, look around, don't know where to go, try to start their own CLIA lab, which is way more work than they need to do. And sometimes people just give up because they don't, you know, apply for a few grants, and just say, in the future we'll translate this to clinical care, but it never actually happens. So I think the low-hanging fruit are the people who have great ideas that don't know how to get to us. I think I'm really excited to be working with you and hopefully with others in laboratory medicine to be able to help those ideas benefit our patients.
And so maybe CARET is actually an apt name. It's kind of buried underground rather than low-hanging fruit.
I like that. Thank you.